the 8 of you here. So once again, good evening to all of you and welcome to the MLB 203 Biochemistry II class. I guess this is the second biochemistry that you are doing, because I think you did what you call biological chemistry in first year. So you have some feel of biochemistry already. So quickly, we'll go over the course outline to see what our plan is for the semester. So we have a semester covering about 14 weeks. Usually, I try to, for us to finish our content within 12 weeks so that we can have at least some 2 weeks to start preparation towards your examinations. So we try sometimes to, some holidays come in, things like that. So to make a way for any of these eventualities, try as much as possible that the lectures cover a period of 12 weeks out of the 14 weeks. So that if anything at all within the period of the semester, we'll be able to finish with everything that we have planned to do. So the first thing we'll do is just have some introduction to the MLB biochemistry course, which is what we are doing now. And then we'll look at components of nucleic acids, that is DNA and RNA. And we'll go ahead to look at the structure of DNA. We will then look at the structure of RNAs, so different RNAs. And we take a look at that process of nucleic acid, or let's say what is involved in protein synthesis, that is DNA replication. Sometimes you have such replication also in cell reproduction. So we'll take a look at process of DNA replication, recombination. So what we call gene expression or protein synthesis, or the production of proteins out of genes through to after the process of replication, you have the process of transcription and translation. Perhaps you would have touched on some of these things in first year, because this is what is referred to as the central dogma of molecular biology. Then we will go ahead and look at what you refer to as gene regulation. So if you are having genes that are being expressed, there has to be a way of regulating these processes. So we'll look at the different forms that genes are regulated, their expression is regulated in the body. We'll move ahead and look at the structure and function of hemoglobin, and then look at some abnormalities, some disease conditions that can occur due to abnormalities in the structure of hemoglobin. We'll look at the process of the synthesis, the production of heme, which is actually the ring structure of hemoglobin, in which you have the ion, metal ion at the center. You look at the synthesis of that, the heme, what we call hemoglobin, which is a combination of a prosthetic part, which we are calling heme, and a protein part, which is the globin. So we'll look at how that heme is synthesized in the body, and also how the heme is broken down. So in other words, we are looking at metabolism of the heme, which is anabolism and catabolism. Anabolism being the synthesis, and catabolism being the breakdown of the heme. So we'll look at that. Then we will take a brief look at the endocrine system. So look at hormones, the different hormones we have, their activities in the body, how they modulate reactions in the body. Take a look at some of these things, the different types of hormones, adrenal hormones, sex hormones, different kinds. And then lastly, we look at biotransformation, what is referred to as drug metabolism. Even though here, no, it's being referred to as mechanism of detoxification. But of course, it is not every time that the metabolism of drugs results into detoxification. In fact, in some cases, it may even result into toxification. So we'll look at such mechanism as the last thing. Assessment, you already have much knowledge about this, that your continuous assessment carries 40 marks, and then the end of semester carries 60 marks. So during the semester, our activities, what we'll do, and all of that, attendance, all add up to 40 marks. And then examination will be 60 marks. That you already know. So this is the plan of the semester. I will share this course outline with you after the lecture. I will send it to the course rep so he can share it with you, so you will have it and follow through and see how we are doing over the semester, how we are moving step by step through this course outline. So far that is what it is that we are going to do. If you have any issues, any questions, the chat is available, so you can write in the chat, so that after everything we can attend to the comments, questions, concerns, anything you want to say, you can write there. Then also, of course, after the lecture, I will open the floor. So if anybody wants to say anything, you can raise your hand, and I will enable your microphone. Then you can unmute yourself and you speak to the hearing of all of us, and we will address your concerns. So that is the plan for today. I want to say that yes, today the link is open for anybody to join, but subsequently, when we succeed in getting your official emails, we will link your official emails to this link, so that it is with the official emails that we will be able to take proper attendance, and possibly also assess your participation with whichever materials that will be available to you. Maybe some tests, things you should try your hands on to have a better understanding, to prepare yourself, and all of that. So we're working on it. I've gotten your lists, so I'll be sending them to the IT office to see if they can send you activation links. And then with these activation links, you can activate your emails, and then uh, we will have a proper house in place. Now, before we start, I want to say that I hear many students complain about online lecture. Is this this is difficult? This is this and all of that. But I think that it is actually one of the best opportunities that you have, that everyone is behind a device that you can actually see clearly what would have been on a screen in front of a large class that some people will be sitting at the back and will not be able to see it well. And another advantage to it is that, for instance, at this moment, this lecture is being recorded. And so in the end, the link is sent to you. And the lecture will be available online. I will send a link. And you can have multiple interaction with the lecture. So you can watch it as many times as you can, take notes. If it was a physical class we were having, you have it that day. When we close, that's it. You won't have access to that again. So it's actually an opportunity. About, I think that right after the COVID-19 pandemic in 2020, there was one student in one of my classes that couldn't attend lectures. I mean, most of my lectures, she couldn't attend because she was sickle cell, and many times she will have crises and be hospitalized and all of that. But she was one of the best students of the class. She actually came to me one day to say, thank you for your video lectures. And she showed me her notes. Said, look at my notes. You would think that she was in the class all the time and wrote everything dito. So it's about yourself sitting down. You have to sit down and do the work. My work is to facilitate your learning, make it much easier for you. This process is like a journey where I take a lead. I'm in front somewhere. Maybe we are traveling to a place like Navrongo from Accra, and I happen to take the lead, and you are behind coming, and I'm telling you that, maybe I call you on the phone and I'm telling you that when you reach Konongo, be careful at this curve. When you reach Kumasi, do this. When you reach Techiman, do that. I give you all the directions. And in the end, it is you that is going to navigate your way through. So I will do my best to give you the direction to facilitate your learning. But you have to do your part. If you don't, you have yourself to blame. So please, sit up. No complaints here and there. Online is this, online is that. Don't be stuck to one thing. Be open to new things. I hope that is clear. Okay, so with that word of, I don't know whether to call it an encouragement or caution, or maybe I should just call it a word of encouragement. You don't be so disturbed about biochemistry, because a lot of times I see how students struggle with biochemistry courses, they always say it's difficult and all of that. But why do you want it to be easy? If something is easy, then perhaps it's not worth it. So just sit up. It's a matter of you spending time. Biochemistry is it's not like some storybook you just take and be flipping through reading. When you do it, you have to do it like mathematics. You have a jotter. As you study, you write, take notes, try your hands on things. That way, it makes things better for you. All right, so I think that's okay. We can start from here. Now, even though you've had biochemistry in first year, which of course would have been the first biochemistry ever in your life, because in the second cycle institution, the secondary school, you have done, but you did their biology and you did chemistry, but you didn't have a combination of the two as biochemistry. So what I noticed is that even in the second year, many of you are still lost in your minds what really biochemistry is about. You just sort of relate to it as some kind of a difficult subject to talk of. So I want to take this opportunity to take you through a little bit of introduction of what biochemistry is. So at least you have a clearer mind on this, and then we'll go into the main business for the day. So we'll look at some introduction of biochemistry now. So if you look at the word biochemistry, clearly is coming from two words, biology and chemistry. And biology, is what? Study of living things. I mean, to put it simply, it to be the study of living things. And chemistry, study of chemical substances and the interactions that they have between them. Also, simple like that. Now you have a combination of this, which means that the interaction of biological substances. So, or you have these biological substances have some chemical components in them. So the interaction of chemical substances in living things or in biological system. So that is chemistry in living things. The study of the chemical substances in living things. So that's what it is. I mean, basically. But let's break it down, go much, to the basics, down, and then we'll find the very ground where you have biology and chemistry meeting. So when you were doing biology in the secondary school, for instance, first, even sometime in primary school, you classify things as living things and non-living things. So the living things, you usually refer to them as living organisms. Now, if you take just living organisms, at the first stage, what you do is you want to classify these living organisms. Are they animals? Are they plants? The fungi. So you have what you refer to as classification. So you have this classification which you are referring to, kingdom Plantae, kingdom Animalia, all these kingdoms, I think 5 kingdoms, each of them will have what you refer to as phyla. And the phyla were broken down into classes, genus, species. You did that classification at that stage, which I refer to as stage 1 biology. And at stage 1 chemistry, you were also doing classification. Remember, we said it is the study of chemical substances and the interactions between them. Now, these chemical substances, what are they? They are elements. And these elements, you do classification of them. That's when you refer to what you call the periodic table. So on that periodic table, you have the elements that are classified into uh, some metals, alkali metals, transition metals, inert gases, halogens, lanthanides, actinides, all of that, transition metals. You did all that. So that is at the level of classification. Now you move down to stage 2. This time around for biology, you take an organism. So before then, you took all the organisms. Then you start to classify them, you put them into the different categories. Now, you take a single organism and you start to study it, looking at what the organism is made up of. So you say an organism is made up of organ systems. And what systems do you refer to? Let's say in humans, for instance, you refer to, say, the digestive system, the circulatory system, respiratory system, etc. And you then look at the organs that are in those systems. And those organs are made up of what? Tissues. And the tissues are made up of what? Some cells. And then you refer to, oh, the cell is made up of what? Organelles. So you draw the cell and show cell membrane, the cytoplasm, which is made up of the cytosol and the organelles in it and all of that. That's at that stage. In chemistry, now you take an element. You say, what's the element made up of? It's made up of atoms. And so you take a look at the structure of the atom. Say it's made up of the nucleus, which is consisting of protons, some positive particles, neutrons, which are neutral, shells that have electrons, negatively charged particles. You study that. That's the atom at that stage. Then you go ahead, the next stage, stage 3, biology. This time around, you have gone advanced. So you begin to look at structure and function of the organs and the tissue. So if you refer to advanced biology, that is university level biology, a lot of times, particularly for you, medical laboratory technology or medical laboratory science students, you do anatomy and physiology. And what is anatomy about? Anatomy talks about the structure of organs and tissues. And physiology will talk about the function of the organs and the tissues. So together, anatomy and physiology is always talking about structure. If you take the heart, then you want to look at the structure of the heart. And then you want to look at the chambers that the heart is made up of, the type of muscles that make the heart. And then you now look at the function, how the blood pumps blood, in the circulatory system. You take a look at those things. At this stage for chemistry, you now start looking at interaction of elements, because you talk about the elements and then you looked at the atomic structure. Now you are looking at how these elements can begin to form bonds. And once bonds are being formed, then of course you now have the formation of molecules, compounds, complexes. You look at these types of interactions in elements or even between elements. Now you go further. You now go for biology to the cellular level. So now you take a cell and start to look at the structure and function of the cell. Remember, anatomy and physiology is looking at the structure and function of the organs and the tissues. Now you have gone down to the cellular level, which you refer to as cellular biology. So you are looking at the cell, because of course now you are having an understanding that if you're talking about the function of an organ or a tissue, and this is actually being made up of cells, then of course the collective structure and function of the cells will make up the tissues and the organs. So we start to look at the cell wall or cell membrane. So what is the structure? What is the cell wall or the cell membrane of a particular organism made up of? You start to look at the component in there. The cytosol, which is the fluid part of cytoplasm, and then the organelles in it. You study that at that level of cellular biology. In chemistry, you start to look at the interaction of molecules, because already you talk about bond formation between the atoms, how complexes can form, compounds can form, and all of that. Now you start to look at chemical reactions, the types of reactions. You do such classification of types of reactions. You start to look at what you refer to as redox reactions. That's oxidation and reduction reactions. You look at the reaction, what is happening, what are the reactants and what are the products. Simply you write chemical equations like something A reacts with B and giving you C, or giving you C and D. I mean, that's simplification of what happens in chemical reactions. But of course, if you study chemical reactions in more detail, there is more to it than just the equation you've written, but it's convenient. But of course, if you study, you see that maybe there are some intermediates that form in the middle, but not so important for the calculations that you do. Then you also look at what you refer to as energy profiles. You remember you studied things like you have reactants that will require something like activation energy to get to a transition state before a product is formed. What are the conditions for a reaction to occur? Is a reaction going to occur in an acidic medium, or it is going to occur in an alkaline medium? You pay attention to these things at this level. Now, finally, biology and chemistry come to meet at a molecular level, because from that cellular level, you now want to see that. So if you take, for instance, a cell membrane, which molecules make up the cell membrane? For instance, so if you take a cell membrane, you start to look at, okay, this cell membrane is made up of some phospholipids, and in them you have some proteins that are embedded in there. You can have some complexes of carbohydrates and proteins, glycoproteins, which are on the surface of the cell membrane acting as receptors. So then you are talking about molecules, which you refer to as biological molecules. And generally, the biological molecules that you refer to all the time will be carbohydrates, proteins, lipids, nucleic acids. So every time you see that the biochemistry you are doing is about these molecules, these biological molecules. So either it's some carbohydrates, or some protein, or some lipids, that is fats and oils, or some nucleic acid. So like today, we come to talk about nucleic acids. That's a biomolecule. So you're talking about biochemical reactions. That is now reactions in biological systems. So then you have your biochemistry. And what are these reactions? It is generally what you refer to as metabolism, because it's a number of reactions that are responsible for the sustenance of the living system. So you want to look at there too, the types of the reactions. They are mostly, it's going to be reduction, oxidation reactions. Well, sometimes you can have things like acid-base, interactions. You look at what are reactants, what are products. You look at reaction profiles, the energy profiles. What amount of energy is required for a reaction to occur? And it is at this point that you start to talk about things like enzymes, which are mostly proteins that are responsible for the speed up of these biochemical reactions. Of course, if you were doing a physical reaction in the laboratory and you wanted to, for instance, increase the rate of that reaction, sometimes you could just easily, heat the reaction system up, to give some energy to the reactant molecules, and you have the reaction running. But this is not an option for biological systems, because biological systems are made up of biomolecules, and particularly proteins and nucleic acids, that if you increase temperature up to a certain point, you have what you refer to as denaturing, something like that. So you have enzymes which are responsible for the reduction of the energy, that is the activation energy that is required for the reactions to occur. And these reactions are supposed to occur in the body in milliseconds. A lot of reactions are occurring in us at all times. As we sit, as you are watching, as I am speaking, I am moving my hands, and your eyes are moving, your ears are listening, all of that. Reactions are occurring. So sometimes I refer to a biological system as a complex chemical system, like our bodies. Many things that are happening. Yes, chemical reactions are occurring in the body to sustain our life. And of course, there too, you look at what is the reaction condition. So for instance, you have some enzyme like amylase, which will be acting in the mouth. There is a particular pH at which the amylase will be active. If it goes into the stomach, the pH there is different. A different enzyme will be active there. So these are reaction conditions. So generally, this is what biochemistry is about. Chemical reactions or chemical interactions, which is generally what you refer to as metabolism. And metabolism is divided into two parts. Catabolism, which is breakdown of big molecules into smaller molecules, or anabolism, which is smaller molecules coming together to form bigger molecules. So, it's not so much of a complex thing. So I hope that this brief introduction will now clear your mind on what really biochemistry is about. So from here, we can move on to the business of the day, which is the component of nucleic acids. And so you see, at all times in biochemistry, when you take any component, or when you take any biochemical substance, or let's just say if you take any component of the biological system, you want to see what that thing is made up of. So you first study the structure of it. Then you want to say, what does this thing do? So you begin to look at the function of it. So you see that at all times, for instance, if you take protein, you first will look at the structure of the protein. And so you want to see what is protein made up of. And you say the protein is made up of some amino acids. you start to look at what's the bond between the amino acids. How many amino acids do you have? How do these amino acids behave? You look at all of these things, the structure of the protein, and then you now see what are the functions of the proteins. You now begin to see some proteins are enzymes. You have peptide hormones. Those are some proteins. Some proteins are structural, responsible for maybe those that are giving some structure to maybe the fingernails, toenails. That's keratin. Collagen, which is giving some cushioning at our joints, things like that. Same way, if you take nucleic acid, because nucleic acid is one of the things that we are coming to study, we are starting with looking at the components of it. What is nucleic? What are nucleic acids made up of? That's what we're taking up today. So let's go into it. So what are nucleic acids? These are we call it a storage of genetic information in cells. So they store genetic information and they transfer this genetic information. So genetic information is transferred from parents to offsprings by nucleic acids. And you have nucleic acids divided into two main categories. You have what you refer to as a ribonucleic acid, which is RNA, and deoxyribonucleic acid, which is DNA. Let's go a step further down. What are these nucleic acids made up of? These nucleic acids are made up of what we refer to as nucleotides. So you can see the trend. It's the same way as we say that proteins are made up of amino acids. The same way we say nucleic acids are made up of nucleotides. Or if you take carbohydrates and you say carbohydrates are made up of monosaccharides. So monosaccharides will bond together to give you bigger carbohydrates. The same way you have nucleotides, which will bond together to give you nucleic acids, which will either be ribonucleic acid or deoxyribonucleic acid. Now we will move a step further down to see what are these nucleotides made up of. What are the nucleotides made up of? So if you take any nucleotide, the nucleotide is made up of three components. You have what we refer to as a pentose, or a 5-carbon sugar, as the central component of a nucleotide, which you find in nucleic acids. And on that sugar, you have a nitrogenous base, and you have a phosphate group. So these are the three components of a nucleotide: the pentose sugar, a nitrogenous base, and a phosphate group. That gives you a nucleotide. Right. Now, what we'll do is we will take each of these components and look at them. Well, for the phosphate group, not much to say about it because it's just coming from phosphoric acid. And so just like all acids behave, they give out hydrogen ions. And so the phosphate group will be responsible for carrying negative charges. Of course, in the aqueous environment in living organisms, the phosphate group that from a phosphoric acid gives out hydrogen ions. and so will be carrying negative charges. So of course, nucleic acids in biological systems are charged. They carry negative charges, which of course explains the reason why you can use practically in the laboratory when you go doing things like electrophoresis, because you have nucleic acids that are carrying um, uh, negative charges. When you apply an electric field, you have them moving towards what you refer to as the anode, or the positive terminal. Nitrogenous base, yes, we will look at it. And then we'll look at the types of the pentose, the 5 carbon sugars that are present in DNA and RNA. As I said, the phosphate group is an inorganic group that not much can be said about it, apart from what I have just said now. So that's okay. So we'll look at the nitrogenous base and then the sugar. So let's go to the nitrogenous bases first. The nitrogenous bases are divided also into 2 categories, what we refer to as purines and what we refer to as pyrimidines. So these are the 2 categories of nitrogenous bases that we have. Now, the purines are 2, adenine and guanine. So these are the 2 purines that we have. These, you find them in both RNA and DNA. Now, the pyrimidines are 3. You have cytosine, you have thymine, you have uracil. Now, if we come to base pairing, you come to see that in the place of thymine, which you find in DNA, in RNA, you have uracil. That's why you have the note written under it that thymine is in DNA only. You don't find thymine in RNA. The same way, uracil you find in RNA only. You don't find it in DNA. I think you did something concerning that already in the first year. So I think even in SS, there was things, because I remember there were some students I taught who mentioned that, yes, they learned something about thymine, uracil, thymine in DNA, and uracil in RNA. So when we come to base pairing, you see that over there. Now, I want you to pay attention to these hydrogens that are shaded in all the nitrogenous bases. You'll see that you have some hydrogens that are shaded. So these hydrogen in adenine, on this nitrogen number 9, here too in guanine, the same thing. Here in pyrimidines is the nitrogen number 1. You have hydrogen on them. Pay attention to those hydrogens, because we'll come to them. You'll come to realize that it is these hydrogens that are the reactive part of these nitrogenous bases that form the bond with the sugar. So we'll see it when we are going ahead. So pay attention to those. Also pay attention to the numbering of the atoms that are in the rings. So you notice that purines have 2 rings. You have a 6-sided ring that is fused with a 5-sided ring, the 2 purines. And the pyrimidines, just a single ring, 6-sided ring. So how you study these things to remember them is that you know that the purines have 2 rings. So if you see any question on the nitrogenous basis and you see 2 rings, 1 6-sided ring fused with a 5-sided ring, quickly your mind should be that this should be a purine. Then what are the purines that you have? You have only 2 of them. These are adenine and guanine. And how do you distinguish between adenine and guanine? You see, adenine is having an amine group on carbon number 6, the number 6 there. If you take guanine, you see that on that same carbon number 6, you are having a double bond O. It's an oxygen rather in place of that amine group. And you have an amine group on carbon number 2. So quickly you can distinguish between adenine and guanine. When you come to pyrimidines, how do you distinguish between them? Certain things are common to the three of them. That is that all of them are having on carbon number 2, double bond, there's oxygen, double bond, oxygen, double bond, oxygen. All of them are having that. So where do you find the differences? Cytosine is having an amine group on carbon number 4. No other of these pyrimidines is having that amine group. So if you see an amine group in a pyrimidine, so first of all, you have only one ring, a 6-sided ring, tells you it's a pyrimidine. You see an amine group, quickly you know this is cytosine. Now, how do you distinguish between thymine and uracil? These two are having a double bond oxygen on carbon number 4. So that is something which is common to the two of them too. So how do you differentiate between them? What you see is that on carbon number 5, there is a methyl group, CH3 for thymine. You don't find that in uracil. So you can distinguish between thymine and uracil. Thymine is having CH3, a methyl group, that you don't find in uracil. And then, of course, lastly, uracil is not having that methyl group there. It's not having an amine group. So quickly, you know that that is uracil. So you can study these things by paying attention to the differences. Because as for the similarities, quickly, they are the base you know for all of them. Then you pay attention to the differences that is between them, and you can easily distinguish between these things. So in exam, for instance, maybe I bring all of these, and you should name them, for instance. You shouldn't have difficulty doing that. Right. Now let's move on to the sugar. We have just looked at the base. So now we go to the sugar, the 5-carbon sugar, the pentose. Now, there are 2 types of sugars that you find in nucleic acid. You have the ribose and you have deoxyribose. And of course, that is where the name is coming from. In RNA, listen to the name, ribonucleic acid, RNA. Ribo is coming from the sugar, ribose. And DNA, deoxyribonucleic acid. So there too, DNA, the name deoxyribonucleic acid is coming from the sugar deoxyribose. Now, if you take ribose and you compare it with deoxyribose, what's the difference? You see, intentionally, you have the differences shaded for you to see. Everything else is the same for these 2 sugars, except that you have on carbon number 2, ribose is having a hydroxyl group, OH. Deoxyribose is having just a hydrogen. There is no oxygen there. So think of it this way. Deoxy means oxygen is deleted. Oxygen is removed. Without oxygen. So on carbon number 2, for the sugar in the nucleotides of DNA, you have deoxyribose. You have just hydrogen on carbon number 2. There is no oxygen there. Deoxyribose. For RNA, ribonucleic acids, you have ribose that is having a hydroxyl group OH on carbon number 2. So these two, clearly you can see the difference between the two. Right. Now we are moving to joining the components. We've seen the sugar, we've seen the base. So how do these two join? First, we also saw that there's a phosphate group there, which I spoke about briefly. Now, I don't know if you studied some carbohydrates in the first year, but see, the same way as you have, say, in proteins, the bond that is between amino acids, you refer to that as a peptide bond. Sometimes some people say amide bond between amino acids to form peptides or proteins. The bond that is between them, those amino acids, you refer to them as peptide bonds. The same way, when you go into carbohydrates, the bond that exists between monosaccharides, you refer to it as glycosidic bond. Glycosidic bond. And so bonds that monosaccharides form with other components are referred to as glycosidic bonds. Now, remember, I told you that pay attention to the hydrogen on the nitrogenous base that is shaded, because it is that portion that is reactive. And so we will come to it and you will see. So have you seen? Here you have the nitrogenous base, that hydrogen is shaded, and you have on the sugar, this is ribose. On carbon number 2 is the hydroxyl group, so this is ribose. And you see that the interaction between that hydrogen, which is shaded on the nitrogen of that adenine, is interacting with the hydroxyl group on carbon number 1 of the ribose to give you water. So water is given out, and you have what you refer to as a glycosidic bond forming between the ribose and the base. That we refer to as adenosine. That's adenosine. Now, even though you see that the interaction between the base, the nitrogenous base, and that sugar is between a hydrogen and a hydroxyl group, don't confuse this to think that it is an acid-base interaction. No, it is not an acid-base interaction. This we refer to as a condensation reaction, because it's giving out water. It's just like how the meteorologists will say condensation, which is like rain. Water is being formed. But why is it not an acid-base reaction? Of course, did you listen to the name nitrogenous base? This is a base. It's not an acid. So of course, that hydrogen which is coming to interact, you cannot refer to it as an acid that is reacting with that hydroxyl group. And the sugar is also not a base. I think practically, if you are in the lab, you take any of these sugars, you can try it out. You go to the lab, you can take common sugar and pour it inside water. and then try to check the pH of it and see if it is going to give you a pH which will be basic alkaline. No, many of these sugars are slightly acidic to neutral. Maybe it give you a pH about 4.55 up to about 7, thereabout. So it is not a base. So the interaction there, keep in mind, is not an acid-base interaction. It's a condensation reaction. Now, remember that at this point, a nucleotide is not yet formed. What we have here now is what we are referring to as what? Nucleoside. So there's a difference between nucleoside and nucleotide. Remember we said a nucleotide is made up of 3 components: a pentose, a nitrogenous base, and a phosphate group. There is no phosphate group on this yet, so it is not a nucleotide. It is a nucleoside. Right. Same way as we took that purine. If you take a pyrimidine, like uracil, it is the same way the uracil will form a bond with, say, a ribose to give you what you refer to as uridine. So here too, that bond there is a glycosidic bond. Great. Now, we can move on and now go to look at the nucleotide, where now you have phosphate group, the phosphate group that is being attached. So you have here, the structure that you see is having 3 phosphate groups. This is actually the structure of a common nucleotide, which you refer to as ATP, adenosine triphosphate. It's a form of cellular energy. But you see, in all nucleic acids that we talk about, DNA, RNA, the nucleotides that you find in them are monophosphates. You don't have triphosphates, diphosphates in nucleic acids. So, for instance, if we're looking at a nucleic acid, DNA, and this is adenosine monophosphate, which ends here, the first phosphate group, just one of the phosphate group this way, one of the phosphate group attached, that is without the last 2 phosphate groups. That is the nucleotide, the adenosine monophosphate that you find in DNA. This is just to show that you can have a diphosphate, a triphosphate, and that ATP, adenosine triphosphate, adenosine diphosphate, they are some forms of cellular energy involved in several reactions in the body. But what you find in nucleic acid is monophosphate. So you see, we talked about a nucleoside, which was having a sugar bonded to the base. So you can actually refer to a nucleotide as a phosphorylated nucleoside, because it's a nucleoside on which you have a phosphate group attached. So you can say a nucleotide is a phosphorylated nucleoside. So now we have the formation of the nucleotide. The nitrogenous base is bonded to the sugar by a bond we are referring to as glycosidic bond. And what is the bond that is between the sugar and the phosphate group? That's an ester bond. In fact, you refer to it as a phosphoester bond. Because an ester will be having C, that's a carbon bonded to an oxygen bonded to another carbon. That is an ester. So in place of, say, one of the carbons, if that's a phosphorus, not a carbon, it's an ester bond, all right, but it's a phosphoester bond. So keep that also in mind, that what you have, the bond that you are having between the sugar and the phosphate group is a phosphoester bond. You will come to understand again when now we are joining the nucleotides to give you a chain, a fragment of a nucleic acid like RNA or DNA. There too will refer to the bond that exists between the nucleotides. So if you understand this as a phosphoester bond, when we mention the name of that bond that is between the nucleotides, you will relate with it. Right. So if we summarize all of these and we take, say, the nucleotides that can be present in DNA and the nucleotides that can be present in RNA, then we will see that we have 4 bases that can be present. We look at 5 bases, but we said in DNA you have thymine, and in place of thymine you have uracil in RNA. So in each of these nucleic acids, you have 4 bases. So if you have in DNA, you have adenine, you have guanine, you have thymine, and you have cytosine. Now, if these bases bind on deoxyribose, they will give you their nucleosides. So you have adenine giving you deoxyadenosine. Guanine will give you deoxyguanosine. Thymine will give you deoxythymidine. And cytosine will give you deoxycytidine. Now, if you now attach the phosphate group to give you the nucleotide, you now have deoxyadenosine monophosphate. Yes, there's a number there, 5 prime. 5 prime is showing that the phosphate group is on carbon number 5 of the sugar. So if you did biochemistry in first year, perhaps you did some carbohydrates, you did the numbering of the carbons. If you didn't, go back and take a look at how the numbers, the carbons that are present in the ring are numbered, and you see that is carbon number 5 on which the monophosphate, because the first carbon number 1, that is what is bonding with the base. So if you count clockwise, you see it is on the 5th carbon that you have the monophosphate. And so you see abbreviation. There's a small letter D in front of it to distinguish it, to mean that it is a deoxy. The sugar that is present in there is deoxy. So deoxyadenosine monophosphate, that is in DNA. And that runs through for all of them. Deoxyguanosine monophosphate, deoxythymidine monophosphate, deoxycytidine monophosphate. If you come to RNA, you have the bases, adenine, guanine, uracil. So remember, in place of thymine, you now have uracil, and then you have cytosine. Here too, the bonding of the bases on the ribose. So this time around, the sugar is ribose. So have you seen the nucleosides up here for DNA are starting, the name is starting deoxy, deoxy, deoxy. But here, it just says adenosine, guanosine, uridine, citidine. It tells you the sugar in there is ribose. And so now if you now attach the phosphate group, the nucleotides that you find in RNA, adenosine monophosphate, there is no D in front of that abbreviation. The sugar there is ribose, AMP, adenosine monophosphate. You also find guanosine monophosphate, uridine monophosphate, citidine monophosphate. Right. Now, apart from these these nucleotides, apart from these nucleotides that we have looked at, which are present in nucleic acids, you can have some other. You may come across some other nucleotides. They are not present in nucleic acids. Just like I mentioned that those nucleotides, the diphosphate and triphosphates, are not present in nucleic acids. The same way you can have things like cyclic nucleotides. And why cyclic? Cyclic because you can see the phosphate group, which is on carbon number 5 here, has looped and formed some intramolecular circular structure with, say, carbon number 3 there. That's why you see in front of it, say 3 prime, 5 prime, because that cyclic structure is formed between carbon number 5 and carbon number 3 of the sugar. And you come across things like cyclic adenosine monophosphate, which will act like a second messenger. I think when we come to the endocrine system, talking about hormones, for instance, you see the activity of the cyclic adenosine monophosphate as a second messenger. So these are not components of nucleotides. Same way you have the cyclic guanosine monophosphate, which also act like some activators of some enzymes, some protein kinases responsible for the transfer of phosphate groups from one molecule to the other. These are cyclic nucleotides but they are not in nucleic acids. So if you see them, know that they are nucleotides, all right, and they are not in nucleic acids. Same way, you have things like 2', 3' cyclic nucleotides. Why 2', 3'? Because the cyclic structure is formed between carbon number 2 and carbon number 3. These usually you find them in the process of metabolism of nucleotides, the breakdown of nucleotides. So some intermediates formed and they really don't stay for long, and then they are consumed by some ribonucleases. I mean, enzymes break this down. When you are having things like recycling of nucleotides, you can have the breakdown of a nucleotide into their components, the base, the sugar, the phosphate group. So there's that, cycle, recycling of nucleotides in the body, just as it is with all other biomolecules in the body. Biological systems are dynamic, they are not static. So you have cycles. Right, now you can have also a dinucleotide. So two nucleotides can actually form a cyclic structure. So you have what you refer to as a cyclic dinucleotide. So like if you take the structure, for instance, you see this, the best way to just take a look at it is don't be scared about these chemical structures. Just take your time, have patience for yourself. Step by step, you look at it. So you take a sugar, this sugar here is having a base on it. What base is that? That is guanine. That is guanine. And it's having a phosphate group on carbon number 5. If you take this sugar, you see this sugar is also having a base on it. What base is that? That is adenine. And it's having a phosphate group on carbon number 5. And you see the phosphate group on this nucleotide is bonding on carbon number 3 of the sugar of the second nucleotide. And the phosphate group of the second nucleotide is also bonding on carbon number 2 of that first nucleotide. So you have that cyclic structure that is being formed by 2 nucleotides. That's why you refer to it as a cyclic dinucleotide. The previous cyclic nucleotides we saw, we just said cyclic nucleotides, but this is a cyclic dinucleotide. It's made up of 2 nucleotides. These two they are not things that you find in nucleic acids. You find some of these as also some second messengers in some bacteria, which is responsible for regulating the things like virulence, the ability of the bacteria to cause harm to its host, the movement or sessility, non-movement of the bacteria, things like that, or the formation of biofilm. That is a protective matrix in which the bacteria could live on a host, things like that. You find them participating in those processes. All right, I think for today, this is okay. I've been talking continuously for more than 1 hour. So it's now your turn. If there is anything, it's now your turn to raise your hand, and I'll call your name. I will enable your mic. You speak, and then I can respond. Or if anything, you can also write in the chat. So the floor is open.